All right, let's move on to our next topic, gametogenesis. Gametogenesis is a natural extension of our coverage of meiosis from the previous lesson. And gametogenesis means the process by which gametes are produced. Gametogenesis involves meiosis, but not exclusively meiosis. There are other processes involved, and that's actually the reason that we need to look at it. Gametogenesis occurs in all sexually reproducing organisms because they all must produce gametes. Thing is, though, that there are significant differences even between males and females of the same species in how they produce their gametes, and when you consider how gametes are produced between different organisms, the differences can be very, very dramatic indeed. We're going to be sticking to humans. Within humans are animals, and the process of gametogenesis for sperm is called spermatogenesis, and the process for creating ova is oogenesis. Notice it's not oogenesis, it's oogenesis. You say the O twice. All right, let's launch into the first of these, spermatogenesis. What I'm going to show you is my summary of the many resources that address this topic. If you go out and look at other material, and I actually suggest that you do, whether it's on YouTube or whether it's a web page on the net, you're going to find that different people emphasize different things and have different levels of detail. What I'm giving you here is my take or my summary of the level of detail that is appropriate for this course, because you can go into much more detail, and of course you can go into somewhat less detail. But let's get down to it, and we will start with the first cell, a spermatogonium. So we are at this point in the testes or atestis of a male, and spermatogonia, that would be the plural, are cells that are dedicated to creating sperm, and they exist in the body of a male before they are born. Spermatogonia are diploid. They have 2 N chromosomes, and they're often considered to be a kind of stem cell. And the first step in creating sperm from spermatogonia is mitosis, not meiosis. Remember, meiosis is a part of this process, but only one. And you'll see there are 2 arrows. The first arrow shows that the spermatogonia are divided by mitosis and create one primary spermatocyte, which is still diploid. But we have this little loop here. And what that's showing is that mitosis, of course, creates 2 cells. It doesn't create one. One of the cells goes to being a spermatocyte, but the other one is recycled, if you like, or purposed back to being a spermatogonium again. Why? Well, there's an analogy which I think might help here. Imagine that you go to work, but it costs you $50 a week in petrol to go to work. Each week you earn $100. Now, you go to work for the first week, the very first week of your job, you get paid $100. You could get to the end of the week and spend all that money. But then how would you get to work the next week? Because you've spent all your money. So what you should do is spend $50 on whatever it is that you need to use it for, but you keep the other 50 to spend on petrol so you can go back to work. So that's what's happening here. The spermatogonium divides in 2. One of them gets sent to be a spermatocyte, which will eventually turn into sperm, but the other one goes back to do the same job again and produce another primary spermatocyte. And so the cycle goes around. Now, this process begins from puberty. Remember that the spermatogonium or spermatogonia are present in the testis of the male from birth. They don't begin at puberty, but they begin changing into spermatocytes at puberty. And in fact, this process, this cyclic create one and recycle the other process, is going to go from puberty until that male dies. All right, well, the primary spermatocyte, which as we have seen is diploid, undergoes meiosis 1 and creates 2 secondary spermatocytes, which you can now see are haploid. Next, the secondary spermatocyte undergoes meiosis 2, and so we have 4 spermatids. They, of course, are also haploid. The spermatids mature, and there's a special name for that maturation process, but we're not going to worry about it. But they grow long whippy tails, and now we have 4 sperm, and of course the sperm are haploid. So although we have lots of new names, lots of new technical words, the process really looks very much like ordinary meiosis, with a round of mitosis at the beginning to generate the first cell. So that's fairly straightforward. And that is spermatogenesis. Let us now turn to oogenesis. Well, things start out fairly easily. We start out with an oogonium. It is a diploid cell. And the oogonium undergoes mitosis to create a primary oocyte, which is also diploid. So far, so good. The next step will be meiosis 1, but at this point we have to pause and look at some very significant differences which are not yet evident. The first thing is that all of this that we have looked at happens in the female baby girl before she's born. Remember that in the male, the spermatogonium goes through mitosis only from puberty onwards. But this process of mitosis, this process of creating a primary oocyte, happens in the womb. Indeed, the first step in meiosis happens within the womb. And there's B for birth, you can see that. But even this first step in meiosis is not completed yet. In fact, the completion of meiosis 1 doesn't happen until puberty. So there is 10 to 14 years in this gap here where this process, where there is no meiotic division, there is no further development in the oogenesis process. All right, well, that's our first difference. The other thing is you'll notice the different sizes, and this cell up here, the large one, is called the secondary oocyte, but this little one is called a polar body. Now, it's got nothing to do with it being cold, and you'll notice that each one of these 2 cells has 1 chromosome, so they're now haploid. Please don't get too caught up in this chromosome being smaller. It's not really smaller. I just had to shrink it to make it fit on the graphic. But the chromosomes are the same size, but the amount of cytoplasm is very different. And so, in fact, almost no cytoplasm gets given to this polar body. It's not going to be used for anything. It may divide, as we shall see shortly, or it could just disintegrate and be absorbed by the woman's body. This one, however, is viable, does have a lot of cytoplasm, and is important. Okay, so we're now at puberty, and the next steps are going to happen to the woman from puberty all the way until menopause. And menopause is the point where she has used up all of the primary oocytes that were given to her or that were created in the womb. So unlike spermatogenesis, with oogenesis, she doesn't get any more. Okay, so we have to go to meiosis 2. And again, we can see something different. So our secondary oocyte divides and becomes an ovum, that's the large one, again, getting most of the cytoplasm. But this process also creates a polar body. The other polar body may divide in two. Now, it depends on which book you look at, which website, which resource. Sometimes that gets left out altogether and you end up, the graphic shows only two polar bodies and other graphics will show you three. But it's really quite irrelevant because all of these three will die, will disintegrate, and will never be used for anything. But what we have achieved is 1, 2, 3, 4 haploid cells, but only one is viable because only one has enough cytoplasm to do the job. The other thing to note, and the reason I've put an asterisk here, is that the completion of meiosis 2 doesn't happen until this ovum, the only viable one of this whole meiotic process, is fertilized. So if we go back here, the primary oocyte goes through meiosis 1, but it doesn't complete it until puberty. So there's a gap, there's a pause there. Meiosis 2 starts, but there's also a pause here until there's fertilization. All right, so we now have an oogonium, which is diploid, becoming a primary oocyte, becoming a secondary oocyte, which is haploid, and then finally a haploid ovum with 2 or 3 polar bodies, which are non-functional and will disintegrate and be absorbed. So I think you can see there are very significant differences between spermatogenesis and oogenesis, and you will definitely need to pay attention, especially to the timing and to when the cell goes from being diploid to haploid. But all the other steps should also be memorized also. Finally, here are your standard resources in Pearson in Chapter 6, and there are the pages. There's a couple of excellent YouTube clips to help you out. I've only given you 2. There's plenty of other ones, but as I say, they differ in their length, in their detail, and how well they're presented. So this is a good start for you, and I strongly recommend you watch them both because this is a difficult concept with many technical terms, and I have simplified it, even though it does look complex. And finally, here are some problems, but you can pick them up from the original PowerPoint to do in your own time.